Namaste to all students. Khushro Bidya, myself Jeevin Dr. Khushbu Verma from Faculty of Agriculture and Veterinary Science. Today we are going to discuss about plant tissue culture media session 2. This session is related to plant biotechnology course of program MSc BT second sem. So before starting, we have to discuss that in the plant tissue culture media, there are two types of major components, which are macronutrients and micronutrients are present in plant tissue culture media, and we have discussed in our previous section. So in this session, we are going to discuss about the remaining component of plant tissue culture media. So third component is organic nutrients, which includes vitamins, and vitamins are basically required for the plant synthesis, and it is essential for many biochemical reactions, and it is used for cultured cell, and basically cells are capable to produce their vitamin at some level but not in the enough level, so they require an exogenous supply of different vitamins for optimum growth. Most usable vitamins are thiamine, pyridoxine, nicotinic acid, vitamin B complex. Next vitamin is a hexitol. Next component is hexitol, and most culture media have this compound. It is essential for the seed germination, sugar transport, carbohydrate metabolism, membrane structure, and cell wall formation. Mannitol and sorbitols are the famous example of hexitols which are used in plant tissue culture media. Next component is amino acid. So amino acid base in the amino acid, glycine is very most common used amino acid in different culture media. It is not essential, but nitrogen containing amino acid enhance the cell growth and plant regeneration process. And if you are talking about the carbohydrate, so cell and tissue requires exogenous supply of carbohydrates to replace the carbon which the plant normally fixes from the atmosphere by photosynthesis. And it is supplied by adding sucrose in media, not glucose. There is a reason behind the use of sucrose rather than glucose, because when you are preparing media, so after preparation of media you go for the autoclaving for sterilization process. So if you are using glucose, so at high temperature and high pressure it will be degraded, but when you are using sucrose as a carbon source, so it will be breakdown into its components and provide the glucose in tissue culture media after autoclaving process. It will not be degraded, it will be breakdown just in in two components. And if you are talking about the concentration of sucrose in media, so per liter it is required 20 to 30 grams. It is required in the highest amount. And next one is the gelling agent. So gelling agent you require is agar agar, and agar is a natural product of seaweeds, and since 1658 agar agar is obtained from red algae, and algae name is Gelidium gracilaria, and with water it melts at 100-degree temperature, and basically it solidify at 45-degree temperature. Agarose you can also use in the place of agar. It is highly purified agar prepared from the Gelidium species of the seaweed. Agarose basically melt and gel at temperature below 30-degree and dissolve through the boiling process. And agarose is much more expensive agar because it is highly purified form of agar. Next component is gel, right, or Phyto Gel. So gelrite is a naturally derived polymer and produced by the microbial termination of a bacterium which is Pseudomonas elodea. It is a low-cost gelling agent and 0.1 to 0.2% concentration per liter required for this component. And there are some examples of natural media which were used before, like synthesis of artificial media. So there was endosperm fluid or coconut milk, fruit material, potato extract, extract of malts and yeast, animal extracts, protein hydrolysates, coconut fruit, orange juice, tomato juice, banana pulp, potato, malt, yeast, fish emulsion, casein hydrolysate, and peptones. These components were used as a natural media for the plant growth. And the next component, which is very much important and very key factor of preparing the optimum media for the optimum growth of plants. So this one is plant growth regulator. So a plant hormone can be defined as a small organic molecule that elicits a physiological response at very low concentration. Plant growth hormones or substance play an important role in the phenotype. It acts as a messenger between environment and the genome. There are two major plant growth hormones like auxin and cytokinin, and third one is a gibberellin. So first of all, we are going to discuss about the auxins. So it is essential for the cell division, cell elongation, cell differentiation, organogenesis, embryogenesis, and callus formation. And if we are talking about the natural forms of the auxin, so it is IAA, indole acetic acid, IBA, PAA, and synthetic form of the auxins are NAA 240. These are the examples related to the auxins. And auxins are required basically for the callus formation and basically for the root elongation or root division. And next component is a cytokinin. So cytokinins basically promotes the cell division. Shoot proliferation means shoot induction you can do with the cytokinins, and it influences the cell cycle. Basically, it is used in the embryogenesis, but it inhibits the root formation. So whenever you are preparing media, plant tissue culture media for the root induction, so you don't have to use about cytokinin in the media. And synthetic form of the cytokinin is 2IP, which is most active cytokinin. And if you are talking about the natural form, so BAP and kinetin are the most natural forms of the cytokinins, and BAP is very much used in the plant tissue culture media for the shoot induction. Third plant growth regulator, which is also important, which is gibberellin. So it promotes the stem elongation, bulb corn formation, and embryo maturation, but can inhibit the callus growth and root induction. And there are some different examples of the gibberellins, so GA1, GA2, and GA3, but GA3 is most common gibberellins. Next one is the abscisic acid. So it inhibits the shoot growth and germination of embryo. It is thermostable but light sensitive. And most common factor, which is pH of the tissue culture media. So you have to maintain or adjust the pH of media in between 5 to 5.8 before gelling and sterilization with the help of dilute NaOH, KOH, or SCl. pH below 5 will not gel properly, and if pH is above 6, so it may be too hard and it is not suitable for the cell growth. So these are the components which are required for the plant tissue culture media. And how we prepare these plant tissue culture media. So first of all, you have to autoclave all the things like glasswares, cotton plug, or if you are using some cap. So you have to sterilize each and everything, scalpel, and spatula or forceps, and petri dish and conical flask, beaker, measuring cylinder, and distilled water. Along with that, distilled water, if you are using double distilled water, that is very much good in comparison to normal distilled water. But before preparing the media, you have to autoclave distilled water too, along with all these uh, components. After sterilization process, you have to let down cool distilled water, because if you are using hot water, so it can interfere the pH, actual pH maintenance. So you have to let down pH temperature of the distilled water, and after cooling down, you use the autoclave distilled water in a beaker, and with the actual measuring, by the actual measuring cylinder. And for example, if you are preparing 1 liter media, so you don't just use complete 1 liter distilled water. You have to use approximately 900 or 800 ml of distilled water, and then add all the components which are required, especially macro and micronutrients, and in the media. And after adding the macro and micronutrient media, you have to mix properly all these components into the media, and then you have to add plant growth regulators in proper amount. You have to standardize by many practices, and you have to add plant growth media. Then you go for the maintenance of pH. After maintaining pH, you have to add sucrose in that media. After adding sucrose in the media, now you have to make up that incomplete media by measuring cylinder up to 1 liter, because we are preparing 1 liter media. So now you have to make up volume of media till 1 liter. After making up media till 1 liter, then again pouring it back into beaker, and then add agarose or agar in media, and go for the heating process by microwave or by the hot plate. After proper heating up, you have to put all media in proper amount in conical flask, and you just seal up with the silver foil and rubber band, and then go for again for the sterilization process by autoclaving process. After autoclaving, when it happens completely, just take all the components, all the conical flasks from the autoclave, and then just let it cool down. After cooling down, your media will be jellified, and completely it will be solidifying, and then you can use it for the transformation process or tissue culture process. So that's how you can prepare your plant tissue culture media, and you can use it for the culturing process or in vitro genesis process. So if we are talking about the key points of this session, so there are some key points like what is the use of plant growth hormones and why we are adding plant growth hormones in the media, because plant cells are having their own plant growth regulators. But the answer is that plant growth hormones are not capable to synthesize enough plant growth regulators or hormones to just to just go for the cell division process or cell proliferation. So they require some additional plant growth hormone in the media. So we just add up some cytokinin and some auxins for the media. And one more key point, like if examiner is asking or giving you some option, especially for the combination or concentration of the cytokinin and auxin for different combination, like if examiner is asking about the callus induction combination of plant tissue culture growth regulators, so cytokinin and auxin concentration must be equal for the callus induction. But if you are going for the shoot induction, so basically try to use only cytokinin, and especially if you are using just PAP and approximately 3 to 2 ml per 1 liter media, you can use it. And if you are just going for the root induction, so basically just use uh, auxin hormone, and IAA is very much IBA is also common, very common auxin plant growth regulator, which is used for the root induction process. And one more thing, whenever you are preparing the media for root induction, just use half concentration of MS media, Murashe school media. And these are some key points which are very much important for this session, and examiner can ask in the question exam, competitive exams. And examples of these growth hormones are also important. Examiner can ask that IPAA is the example of auxin or cytokinin, or 2,4-D, what is the full form of the 2,4-D, and what is the full form of NAA, what is the full form of BAP. And BAP is the example of cytokinin or auxin. Examiner can also ask that for the embryogenesis or organogenesis or cell differentiation or cell elongation, auxin is very much important or cytokinase is very important. And if you are talking about the gibberellin role, so examiner can ask that for the bulb core formation, which plant growth regulator or hormone is very much important, gibberellin, cytokinin, or auxin. So you have to give answer that gibberellin. And which is the common gibberellin? So GA3 is the very common gibberellin example. So this is the sum up of this session, and this session is powered by digital version 2.0 Jyoti Vidyapeeth Women's University Jaipur. If you have any query, then mention in the comment box. I will resolve it. Thank you.